Okay, so we are home and we have everything ready to go. Copper sulfate that I don't need anymore, still there on the stove burner ripe for a tragic accident. Adjust my mic a little. Okay, and Santitas corn chips, because everyone should have those in their lab area. The only thing I'm missing is Mountain Dew. The laptop is ready to go. The camera, I have cleaned the image sensor, and how I do that, just very quickly, is with usually a dry Q-tip, a dry clean Q-tip, very gently over the CCD, and then sometimes if I have to, I blow off with a coffee stirrer stick. And generally speaking, it never comes to that, because just, using one of these and then a quick inside the camera lens body without the stick is enough to take care of any little speckles. And you can see I did a fine job this time, not a single speckle. Now, that goes in there, and sometimes just the very act of doing this will add debris that you then have to sweat. Not this time, apparently, thank God. That goes on there. It's a little bit loose. The samples are here. I've got pipettes, and I need a slide, but getting a bit ahead of myself here. But it doesn't hurt to have the slide ready. I'm just going to set it where somewhere where it won't get all dusty. So just swipe that clean and lay it there, and it's ready to go. I also need to grab some cover slips and the rest of the stuff to show you how to do wet mount slides. But before I do that, I want to show you something. Got the 3 specimen jars there. What you want to do to see if you can see anything really fascinating before you get into the scope is turn out the lights and then turn on a flashlight, a bright flashlight behind the jars, and lean down and just have a look at them. And a lot of the time, if you got a really good sample, you're going to see all kinds of little particles moving under their own volition in there. There'll be Daphnia, Hydra, large parameciums, perhaps amoebas, juvenile insects, and larval stage things like, crayfish and etc. Very, very cool thing to do, and it can be fascinating all in itself. And now you can see, yeah, I did get a couple particles here, here, and here on the CCD. We're going to ignore those. It's not that big a deal. I'll just remember them as dust particles and try not to imagine them as bacteria. Anyway, okay, next step. Show you how to do a perma slide, what I call a long duration sealed slide wet mount. Okay, now this is a technique I've been wanting to show off for quite some time. I developed this many years ago. Geez, I don't even know how many. But a long time ago, I discovered that a simple idea worked very, very well. What you need is a clean slide, a clean cover glass, your sample, some petroleum jelly, and a toothpick. And now, cheap pipette for your sample. Sometimes eyedroppers or pipettes don't really seal very well, so you go and dip your sample, and before you can even get over to your slide, it's dripping everywhere. There's a trick for this. See, this is loose, and it's going to pull air. It's just inevitable. So what you do is you take either oil or water and drip it down so that it contacts here, and it makes a actually surprisingly effective seal. So we'll do that real quick. See, got it down in there. Spin it to keep it. These are really loose, geez. Partially off, I think. It was really the cheapest eBay had to offer. Okay, so which sample do I want to use? There's a lot of sediment in that one, but it doesn't really talk to me. I want this one. So we'll open that up. Get the petroleum jelly ready, because this is all got to be done quickly. Reasonably quickly. Take your toothpick and get some petroleum jelly on it. And what you're going to do, I should probably seal that vial because it doesn't need to be open just yet. There. Take your slide and your toothpick, and what you're going to do is paint yourself a line more or less the size and shape of the cover glass that you're going to put over it. Keep getting yourself more. And I thought of a really cool idea to make this easier, but I'm not set up for it. You could use a hypodermic, a syringe and a hypodermic needle. Actually put some petroleum jelly inside the syringe and squirt it in a very fine line with your hypodermic needle and draw yourself a perfect little sealing sort of ring. This way works, but it's a little hit and miss. But see, basically, that's what you want to do. And what you're doing is you're making yourself a gasket. Now with that done, you want to take your sample jar, or vial, I keep calling it a jar. Take your cover slip and get it back where it belongs, up there in easy reach. Take your pipette, and I'm going to bail out some of this liquid because when I stick the pipette down in it, it's going to displace. So just spray that on the counter, wherever it'll fall. Go down in. Ooh, God, getting it everywhere. See the problem with that? Alright, I'm stirring up the bottom sediment, but that is actually a good thing. So, get the pipette down in, and go for the crap in the bottom. Let's stir that up a little bit. Okay, there's my sample. I got some sediment and I got some fluid, which is a very good thing. You don't want to fill that area up. You're not aiming to give it a ton of fluid. You're aiming to give it just enough, because when the cover slip now goes over that, it's going to squash that drop. And if you gave it too much, it can kill your seal. That's not what you want to do. You want it to hit and seal, and it's okay to very carefully stick it down. And now you have this. It's actually a sealed slide. If you do this right, this thing isn't just good for minutes or hours. This thing is literally good for days. I did slides like this, bacterial samples, that were actually still integral, still sealed after months. Of course, they were very dead because there's only so much nutrient within the fluid of these slides, and so the organisms use that up, and then they either go dormant or they die outright, and bacteria will consume and dominate the slide and annihilate anything larger. And then all that's left is a lot of bacteria, which eventually go dormant or die. But there you go. Sealed slide, a la Rick. These are awesome. These are also not great for extremely high power use because, you work with very fine distances. It's easy to crash into the cover slip. But if you're very careful, you can use this well. I've done it with bacteria under oil immersion, and you just have to be careful how far you push. Go until you get a focus, which is right at the cover slip level, and just don't push it any further. Okay, shall we have a look and see what we got? Yes, we shall. This is what clamps the slide in. So, one-handing this all the way, you just push, get that more or less in place, and let her go, and she takes care of herself. Nice and neat. There she is in the scope, loaded, ready to go. And there is the view. We are on the lowest power. And presto, we are looking at weird stuff, Holmes. Okay, there, whoops, there near the center, out of focus. Oh yeah, hello. Rotifer, I think. Some protozoans, a little bit out of focus. I need to find a clump that's happy and alive, and then we'll zoom in. Here we are. This microscope has stage controls for forward and backward, left and right, and fine focus, coarse focus, and focus for the condenser itself. Focus on the condenser is very important. That is what maximizes your contrast, clarity, and the lighting style of your specimen. Got some... There's not as much on this slide as I was really hoping there would be. A lot of debris, a lot of dead stuff. I need an interesting target. And there aren't many to choose from. The last time I got a lot of diatoms and a lot of very fascinating organisms, amoebas, certain types of protozoans, rotifers, blue-green algae, amazing stuff. It was a great thing. This time seems fairly boring, actually. Okay, let's kick up the power a notch. I have to refocus because of the change in power. Don't have to be real careful. There's some diatoms. Let me change the resolution because I know this thing went default, which is crap, and that's accounting for a lot of my lack of clarity. So we want the high def setting, and bam, that's much better. Am I right or am I right? Yeah, much better. There's a diatom slowly working his way out of the field, focusing on the crap spot, sediment patch. We got more diatoms. Quite a lot of diatoms. Got some kind of video corruption happening there. Some programmer or another is messing with it. Either that or it's that loose cable. I have a loose cable on the camera inside the body. Need to do something with. Yeah, there's some protozoan activity there. Hard to keep him in focus. He's a fast mover. A lot of diatoms. They just weren't showing up because the clarity wasn't the best. Yeah, there's quite a few there. See? The samples are very much alive. That lake is full of stuff. It's amazingly rich as an aquatic environment. Best I've ever had access to. Of course, also the best microscope I've ever had access to, so how would I really know? There are some other things, mostly diatoms in this. There you can see some to the lower right as well. Some moving, some probably dead. Fast protozoan just went zinging by there. Will you please stop messing with me? Bad connection on the camera, I'm sure, because it didn't do this before. So, oh, hello. What are you? Come on back. High-speed chase. He's moving fast. I can't keep up with him. Who knows where that went to. Clearly an interesting slide. It'll do. But yeah, hey, pretty cool, huh? There you have it. One thing to note, Ooh, one of those long, thin, thready bacterias, I think. In closing, I want to say a word about identifying what you see under the scope. What I do, and of course I am a YouTube author, so I'm there a lot anyway, I use a combination of YouTube and Google. And I just kind of go with my intuition. If something looks round and spiked with hairs, I tend to think, okay, protozoan, go from there, put in a blind search, see what I see out there. And if something looks familiar, close to it, I think, okay, pursue that thread, and just keep going until I come up with what I think is the same species in a photograph, and usually the name of the species will be attached to it somewhere. And so you can get a good idea of what you're looking at that way, and maybe get a positive idea on it. And, that's interesting. I want to get that under high power and see what the heck that is. Also, you might just go ahead and set up, enough of a system to put up some video of your own on YouTube and say, hey, what is this? And let, the public answer for you. You can learn a lot this way. And most people won't mind. You'll get, the usual fools that'll just drive you like crazy with their crap. But the most of the people are cruising around, they're looking for this kind of stuff. At worst, I found you. At worst, they'll say, wow, that's really cool. Can you please put up some more videos? Or how do I do videos like this? But a lot will say things like, I know what that is. That's very obviously, most people will say it's very obviously like you should have thought of this before, such and so species. This dude is really interesting. That's, I'm guessing, some kind of rotifer, although really strange organism. It could also be a larval something or other. Don't know. Anyway, I hope you got some use out of this. I hope you learned a little something from it. I hope I didn't bore you to tears with my endless narrative. And I hope you thought this was as cool as I did. I've never seen one of those before. And I've barely scratched the surface on this slide. Much more to go. So anyway, yeah, hey, thank you for watching, and I have a couple more projects in mind that I want to try out. My battery's about to die, so I got to go. Thank you for watching. Please tune in next time, and see you later. Bye for now.